Welcome. Welcome to our first demonstration video. This time it's on statistical testing as part of our course on exploratory data analysis. So here we're looking at microbiome data, and what we're thinking about is that we have different cases and controls, and what we're going to do in terms of our technique of percentile normalization is map the position of each cases onto our distribution of controls to get the percentile for each individual case. So that can mean if someone is very, very tall, and it is mapped onto a distribution of random people's heights, they're going to have a small percentile because they're quite at the extreme, and so that percentile might be something like 0.01. And then if you have someone who's very small at the other extreme, and it's maybe at the might be at the other extreme of percentiles, like 0.95 or something like that. So we're trying to get the cases data and get the percentiles of where they lie in the distribution of controls, which means that we take the percentiles and analyze that. We don't actually need the data directly from the controls, but we have therefore normalized our cases based on the controls. And this, in the context of microbiome data, could be for a particular taxon of interest. And we do this for all our taxa across all our studies. And what we can, an example of this would be that we want to compare, I don't know, the relative abundance of Bacteroides fragilis after CLR transformation or using the relative abundance. So what is useful about percentile normalization is if you take the raw abundances, our rate of false positives will be very, very high, and this is a bad idea. If we use other methods like COMBAT um, and limit to normalize our data, that does reduce the rate of false positives, which you can kind of get an indication of from this y-axis here, but it's not as well as percentile normalization, at least based on this paper. And having abundance data, relative abundance data in biological contexts is very, very common. Um, so what we can think of it is that we have two distributions here from study one, where we have the cases in blue and the cases in orange and the controls in blue. They have particular distributions where we can see more or less that the for this particular taxa, the case levels of it is higher. Um, but the medians for each data and variances are somewhat different, and therefore we need to put the cases plot them with the controls, so that we can place them on the distribution here. We can kind of see where the cases fall. Most of them are at this high percentile end. And therefore, when we merge our data, it is more possible to compare and merge studies one and two and the associated data than to analyze them separately, especially at the start where there was uh, measurement confounding at the start. For example, the number of counts from the sequence data was inherently different. So this can mean that we can apply this with R code. So now we're going to switch to our RMD file, which you can open up from the download from the Google Drive and examine it there. So what you can see here is that we have several packages that we need to install. So key ones in tidyverse for data manipulation, patchwork is for plotting, per is for also for plotting, and coin has data inside it and functions that we're going to use later. So these should be relatively simple to install. You may need to use install.packages first to install them properly. What we can apply then is you can assign colors to study A and study B as per the plots there, where one is blue and one is orange. This just makes it a link. And then when we run this, we'll find the study colors is just a simple vector with two elements containing those colors. What we can also do is create a function to percentile normalize this. So obviously applying that property is not straightforward. So what is being done in this function? We're defining the function with the word function, and we've got our cases and controls. And what the map DBL function doing it is looking at the placement of each of the cases in the distribution of controls and find the fraction of controls that are more extreme than that particular case, which gives us the percentile. So this is a small but useful function for doing that normalization. And we can take simple, small examples of data where we have a set of control samples here for studies A and B, and a set of case data for A and B, and use the cases to the controls to normalize the cases in this instance. So it uses the function percentile norm to normalize the cases based on the controls. So these are sent up to this function here. They become the cases and controls here, and we obtain a vector containing the percentiles for them. So let's look at what's inside there now and apply these functions to see what's there. Well, we didn't define the functions. That's why it doesn't work. So we define the function, we won't get that error. And then we run through, and what we can see is that we get the new percentiles. So you can see if I zoom in a bit, uh, PCT small, that the new percentiles are different. So 0.04 becomes 0.2, 0.09 becomes 0.8, and that's because um, the the case, that's the controls. The cases are 0.06, 0.25 because 0.25 is bigger than nearly all 4 out of 5 of the controls. And same for the group B, we get their percentiles. So we can then apply this to a larger collection where we have 200 controls. If you want to make this um, more consistent, you can use the function set.z to be consistent with our input data that is used each time. So therefore, even though we've got a random number generator here, each time if we have this on, it'll generate the same set of random numbers. If we change the value inside here, those numbers will change. What the function Pmax is doing here is preventing us from having negative values. So it makes sure that the minimum value present is at least 0. We can't have a value that's lower than that. And what we're doing is generating two other random numbers with a mean of 0.12 and 0.06. And that will create our data to assess here. So if we run through all of that, we find we've created a large amount of data to analyze in the same way as we did our example data above, and we can map the cases onto the controls to get the percentile normalized values for each of them. And we can check the structure to see the cases and controls in the original data, and we can also look at the normalized versions as well too. And you can see that they're all between 0 and 1 because they're percentiles. A logical next step in terms of our plan for exploratory data analysis is to visually plot them and also get a numerical summary for them too. So what we see is we can get a summary stats for each of the controls in the original study. And what we can see for A and B is that their controls have fundamentally different distributions, different ranges there. You can see that straight away. The mean of the first one is 0.12, the mean of the second one is 0.22. For the cases, you can see that their underlying distributions are also different. The median for the first one is 0.09, the median for the second is 0.117. And their interquartile ranges are quite different as well. These are mutually exclusive here. So they're really different. Whereas if we look at the normalized data, what we see is that the medians are now much more comparable. One is 0.265, one is 0.302. And that means much more logical to compare these different distributions. And we can do this visually. So what we're doing here is we're creating a histogram using that data, and we're adding things into it. So we can start with a simple one and just do hist control A large. You don't need to do the copilot prompts. And you get a histogram like that. And that's fine, but it's not very good. So that's why you add the other things in here, like the colors, just to make a nice color. And then we can run that line and some colors, change the number of breaks. So you can toggle the number of breaks to be whatever works for you, but this is the number of bins, and the function will moderate that a bit. So it won't be exactly 22 bins. It'll be some sort of number that works effectively. If we just have 2, it's not the best. If we have a lot of breaks, it's maybe too many as well. So there's kind of happy median there. And then we put on the X labels and the limit the X axis as well for given the distribution of our data. And that creates a plot like this. So what we can do is use the par mf row to plot top and bottom plots, so we can map the controls onto the one from study A from study B onto study A. So we've got the add equals true at the end of this. So if we do that for study B, you see that the controls um, have a different ranges and variances and everything compared to study A. And if we do this for parameter over both, look, we can get this plot. If I widen my panel, so if you see this and it's very narrow, just widen the panels a bit, and you see it a bit better. I need to toggle the Y lim, I think, a bit here because of the binning. I think it's C 15 is probably too many. Let's try it again. 20 might work actually. That's a bit better. Yeah, so there's a high peak there for that particular data set. Um, yeah, because you can see that better. So you can see that the distributions are different. The B is shifted compared to A. So now what we can do is compare the controls and cases in study A in the same way. If we do that, we can see that the controls have a slightly different range to the cases, very slightly, and the same in study B, but the relative abundances of each are different. So there's fundamental differences between A and B that are in addition to our differences between the cases and controls, and we need to avoid the cases and controls confounding what we have. So what we can do is then plot our normalized data. So you can use top and bottom plots again, and we can plot the study A and study B. And what you see is that there's a wide range of data points. There's no intrinsic distribution, which is very, very common for biological data. And what we can do then is test our data statistically. So if we're looking at this kind of data, we're doing a bit of EDA, but we might want to do a statistical test. What we might consider is that the distribution of cases and controls are equally interspersed in terms of the relative abundance of this particular taxon, which would mean that uh, the the average percentile will be 50%. And therefore we can compare our data to a median, which is the average median, of zero 0.5. So we're using a two-sided test using the Wilcoxon signed rank test on these ones compared to a median of 0 0.5. And we find that, yeah, it's quite different from 0.5. It is heterogeneous, but it's definitely different from 0.5, and the same for B as well. So that's one way we can test A and B. A nice thought then is what if we want to compare A versus B. So again, we can use Wilcoxon's signed rank there to apply that, and we get the W1 vector from that containing our results. And what we can see is that we get a W statistic and a p-value. Another way we can compare A and B in this example, where we have no underlying distribution, is to compare the W statistic there using permutation testing. So this means we permute the labels of A and B, their normalized percentiles for those cases, many, many times, and see if there's any real difference between them. This does not assume any underlying distribution. And you can take the difference in the statistic is probably best, or the means or medians, depending on what you're doing. So what we can do is create a large vector containing the scores. So we're mixing these all together. So you have 200 scores inside here. We can check that in the prompt, and we see it there. And what we can do is add on labels. So we have A and B iterated many, many times in our labels vector. And you can see that there. So these are 400 labels of A and B. And what we can do is loop through this loop where we map for each of the for each of the number of permutations, we have a function here that samples across the labels. So it samples the labels, adds them to some vector called shuffled, and then extract the scores that are labeled A and compares them to the ones that are labeled B. But this is entirely random and should give us a null output at the end. And um, this gives us a W stat that is put into the perm stats vector that we can look at the end. So we're going to loop through this, and it shouldn't take too long. but it might on some computers. Now, if we look at the output from perm stats at the very end, that will give us a vector of W values that we can plot, and we can plot our observed statistic onto that. So what we're going to do is create a histogram in gray with the permuted stats, and it'll look something like this. And then we can, using the appline plot, we can map where our statistic falls onto it. So putting a red line there, a red vertical line of where our stat is, and then we get the area under the squares that is more extreme than that particular value, and that's our permuted p-value, which is 0.094. So um, that kind of tells us about how extreme our p-value is. So you can see that the um, permuted p-value and original statistic value are relatively similar. This is one-sided, this p-value. Please note that. And is nice to learn about alternative ways to interpret data. And permutation is very, very robust and good for heterogeneous biological data. So that kind of concludes this part on looking at um, statistical testing using permutation testing, having percentile normalized our data, and we'll continue this in other videos.